Welcome to the Organization of Biological Field Stations live from the field event today. My name is Kerry Wininger with Sonoma State University's Center for Environmental Inquiry. We encourage you to view past recordings and learn more at thevirtualfield.org. For instructors in the audience, be sure to visit thevirtualfield.org to access supplementary materials for your students, such as an instructor guide for live from the field events with suggested assignments and an event resources document full of publications about the specific research topics and projects being discussed today. And I will let Jarrett Byrnes take it away. He's an associate professor of biology at the University of Massachusetts, Boston, an affiliate faculty at the Shoals Marine Lab, where he studies kelp forests and climate change.
Thank you all for joining live from the field. We welcome classes, faculty, and other participants from across the nation. This is the third live from the field event, and in this series, we take you on virtual field trips to research sites around the country and the world. As you'll see today, we are visiting 3 biological field stations, places where people from all backgrounds come together to study the environment. Today, we're traveling to field stations in Central Oregon, Southern Maine, and Northern California to talk with researchers studying climate change at the land-sea interface. The temperature of the land and sea across our planet is changing. Now, these changes in temperature can not only directly affect organisms living in the ocean, but they can have sometimes unpredictable cascading consequences through the tangled web of life in the sea. Our researchers today are trying to tease apart the effects of climate change on ocean life to understand the future of our changing world. Each researcher has prepared an 8-minute video outlining their climate change research in the Intertidal. After the videos, they will all be available to answer your questions live. I'd like to encourage everyone to post your questions in the Q&A as we go along. And students, remember to type student and your full name in front of your question. All right. So let's start with the Shoals Marine Laboratory. The Shoals Marine Lab, a joint institution of the University of New Hampshire and Cornell University, is located on Appledore Island in the Isles of Shoals in southern Maine. This beautiful, isolated 100-acre island serves as the center for experiential learning, modeling environmental sustainability, and wonderful place-based research, which, I'm a little biased as I teach underwater research out there, so I can fully attest to that. Dr. Jen Dijkstra of the University of New Hampshire is an undergraduate research mentor and researcher at the Shoals Marine Lab. Her research centers on understanding the implications of seascape change for the Gulf of Maine. She's attempting to document firsthand the changes in animals and algae on the seafloor due to climate change.
Good afternoon. I'm Jen Dijkstra, a research assistant professor at the Center for Coastal and Ocean Mapping at the University of New Hampshire. A lot of my research is centered in the Gulf of Maine, which is shown on the upper right-hand side, and I work on questions related to how marine communities, like the ones you see here, respond to climate change and how invasive species affect the functioning of marine ecosystems. My research tends to involve a lot of scuba diving, which is really quite a lot of fun, to do field experiments in combination with laboratory studies. The world's oceans are warming very rapidly, and the Gulf of Maine is no exception, and is in fact warming faster than 99% of the world's oceans. You see that here in the bottom panel. The legend on the right indicates temperature anomalies, with red indicating temperatures that are above the normal and blue indicating temperatures that are below the normal. And what you see here in the Gulf of Maine, which is where that little black box is located, and it's very red, indicating the temperature anomaly is much greater here than in other locations. It is also a hotspot for biological invasions, where we have invasive species in both the intertidal and shallow rocky subtidal systems. And you can see that here in this collage of species where we have on the upper left-hand side, it's an animal and it's made up of several colonies. There's seaweed species, there's a few periwinkles, anemones, and of course the dreaded green crab. The questions that I ask are, what are the effects of ocean warming on marine communities? And how do regime shifts alter community architecture and food webs? So myself and my graduate students study marine communities in the Rocky subtidal, and particularly seaweed communities, as they provide habitat to many other species, including fish, and this is a school of pollock that you see, seals, this is a cute little harbor seal, stripers, as well as the cormorant, as well as green crabs and lobsters and other species of invertebrates. One of the ways I look at ocean warming effects is through long-term monitoring studies, where I collect underwater video footage and still images of the seafloor. Here, we are swimming above the seaweed community for the purpose of collecting underwater video footage so that we can stitch it together to create a larger photo mosaic of the seascape. In addition to these images, my graduate students and I collect data on invertebrate abundance using quadrats we set on the seafloor, and we look for lobsters, crabs, and sea stars. I'm also part of the Shoals Marine Lab Intertidal Monitoring program. This is a program that has been going on since the 1980s. This program, in which undergraduate students spend 3 weeks on Appledore Island and collect detailed information on the intertidal.
Why study the intertidal? According to NASA's Earth Observatory, the Gulf of Maine is warming faster than 99% of the world's oceans. The intertidal is a region of importance given its high biodiversity and susceptibility to human impact. It is a region shallow enough to receive sunlight and complex enough to serve as a safe haven or estuary for young marine offspring. It plays a part in oxygen production, a healthy fishing industry, and a stable ecosystem both near and offshore. This region is complex in its ecology and in its issues. Studying the intertidal in the Gulf of Maine can be challenging. It must be done at low tide, in calm seas, and has seasonal restrictions given climate. Methods have been standardized and studied each year by the Northeast Coastal Field Station Alliance at a dozen locations ranging from the Isles of Shoals in Maine all the way up to Bon Portage Island in Nova Scotia. At Shoals Marine Laboratory, there are 26 discrete transects circumnavigating the island. Each year, a group of intertidal ecologists and undergraduate researchers spend several weeks collecting the data to measure zonation and species present along these transects. Over time, we have developed a long-term monitoring data set to measure change in the intertidal environment. Combined with the rest of the stations along the coast, we can gain a comprehensive understanding of the broader intertidal ecosystem in the Gulf of Maine. But how do we do it? First, we need some tools. Keep in mind, this is an inherently wet and slippery environment. Assume that anything going into the field will get wet. Before we head out, we will grab our data sheets, transect tape, quadrat, some 5-gallon buckets, and laminated ID guides. In the field, the first step is to find the transect. Over the years, the intertidal group has used spray paint and eye bolts to mark the location so it can be replicated each year. Once we have dug through the ascophyllum and found the transect, we can lay out the transect tape perpendicular to the water. A quadrat is used to subsample the area along the tape. It would take far too much time to sample the whole area, so we subsample to come up with a reasonable representative sample across the transect. Another method is to randomize the subsample by tossing the quadrat. This serves a slightly different purpose because it will not magically land along the transect. This randomization can help to remove a natural human bias from the sampling. Once the quadrat is laid out, we can pick through it to find sessile and mobile organisms. It is important to spend time moving algae to ensure all organisms are counted. If an organism is too numerous to count, such as barnacles, an X is used on the datasheet. A photograph can be taken to study the quadrat later and store the data. Another variable to assess is percent cover. Percent cover is measured by estimating what percent of a quadrat is rock versus barnacle or species of algae. We could expect the percent cover of the zones to be different as the distribution of species changes across the transect. The spray zone may be 100% rock, while the lower intertidal may be 100% algae. Here's an example of a quadrat in the lower intertidal. Compare it with one in the middle intertidal to that of the upper. Looking at percent cover over time can tell us how the zones have shifted. Transects are a useful tool for collecting information over time in a standardized fashion to accumulate a powerful data set in studying the intertidal ecology of the Gulf of Maine.
All right. Welcome back, everybody. The Hatfield Marine Science Center serves as a base for researchers from Oregon State University and 6 state and federal agencies. It's a gorgeous facility located in Newport, Oregon, with access to iconic rocky intertidal, harbor, bay, and other habitats. There, researchers explore the diverse impacts of climate change in the sea. Dr. Sarah Gravem works out of Hatfield as a research associate at OSU for the Partnership for Interdisciplinary Studies of Coastal Oceans, or PISCO. Her work focuses on the dramatic impacts and recovery from the climate-associated sea star wasting disease that recently swept through the west coast of the United States.
Hi, my name is Sarah Gravem. I am a research associate at Oregon State University in the Partnership for Interdisciplinary Studies of Coastal Oceans, PISCO. It's a mouthful, and I study marine intertidal ecology. So I study how organisms interact with each other and interact with their environment. And one of my main parts of my research is studying how climate change is affecting all the little critters that live in the tide pools. Well, on a fieldwork day, I get up at about 3. And I eat a big breakfast and I drive out to the coast, get here by dawn, and spend 5-6 hours out hitting that low tide to get as much fieldwork in as I can before the tide chases me back out and I can't access my research subjects anymore. So here we are in the rocky intertidal. This is the Intertidal Zone along this transect line that I've laid out. And so right here next to the water, we have the low zone where you see tons of different types of algae. There's some sea stars, sponges, sea anemones, sea urchins, just tons of different organisms all living together. And as we go up the zone, you see these guys, these big clam-like things, mussels, taking over. They are the bullies of the intertidal. They grow fast, they settle in the millions, and they're big and tough, and they kind of push everybody else out. As they take over, they change the whole makeup of the community that's below our feet. And as we get higher and higher and higher, you can see that the mussels disappear toward the top. And that's because it gets super hot in the daytime. So what I'm studying here is how climate change is affecting all the animals that live in this intertidal zone. And so what you can see is that those mussels, for example, have this low zone boundary here, and above us they have a high zone boundary. And so what we call that is a zone, and that's, the mussels are pretty much in the mid zone of the intertidal. And so at the bottom, they are cut off, and at the top they are cut off, and that is their entire like ecological niche. You can walk from one end of their ecological boundary, their range boundary, if you will, to the other end in a matter of steps, which is super duper cool if you're studying something like climate change and how it affects species ranges. So this is a sea star or a starfish. It's called an ochre star. This is our common sea star on the west coast. They come in orange, purple, red, brown, and if you ever have been to a west coast tide pool, you probably have run into them. These are actually voracious predators that eat all sorts of stuff. They're the top predator, and they're so important that we actually call them the Keystone Predator here. And the reason for that is because their favorite food is mussels. And you can see this guy is actually eating a mussel, or just ate a mussel. It is spitted out, it's all digested, and that's just the shell left. And so these guys kind of live in the lower zone. So we just covered how at the top of the zone, there are all these stressors pushing mussels down, and at the bottom of the zone, there are predators pushing the mussels up. And what I'm interested in is how this ecosystem is gonna look in 10 years, in 50 years, in 100 years, and how climate change might change all the zones that we see here. So, for example, if we're talking about mussels, warming climate could do a couple things, right? First, it can push down the upper limits, right? So you've got warmer air, more heat waves, more stress, pushing them down so they'll die at the top and survive, say, in the middle. We also have sea level rise, which is going to make the whole intertidal zone go up, which isn't my expertise, but certainly something we think about. And then at the bottom of the zone, you might think, oh, climate change shouldn't be a big deal down there, right? Because that's the less stressful part. That's the part they're like in the cushy ocean. But the lower limit of the mussel bed is the same, essentially, as the upper limit of their predator. And so whatever climate change is going to do to this predator will affect the lower limit of the mussel bed. So the way that we measure how communities are changing over time is pretty actually low tech, at least on its surface. So this is a transect line. I have bolts that I drilled into the rock, and they're from the low to the high, so I can place this tape at the exact same location every year, and I've been doing this for 5 years now. And along this tape, we can use quadrats, and we use photographs, and we use all sorts of other things too, but we can just lay this down in the same spot exactly every year, take a photograph, and then analyze how many sea urchins, what's the cover of algae, how many mussels, how many sea anemones, and we can use this to look at how the beds are moving down, or biodiversity is changing, or the numbers of sea stars is increasing or decreasing over time. We also look at how the environment changes, and so this over here inside this little cage is a temperature logger. So this is actually recording temperature all year, every 15 seconds. And that's important because the temperature in the intertidal changes really drastically. You go from being underwater and being at like 8 degrees Celsius, 10 degrees Celsius, to on a hot day, this can get up to like 20 if on a really, really hot day. And these organisms that live here have to deal with this extreme fluctuation in temperature, like at least seasonally and often daily. But there's one other big way that climate change is affecting these systems, and that's through disease. And so sea stars, these guys, actually in 2014 here in Oregon, went through what was a sea star wasting disease epizootic, which is epidemic for animals, zoo. This is the largest marine disease on record, we think. The wasting disease started in 2013 in California and Washington, and over the next couple years spread all the way from Mexico to Alaska. The death rate in Oregon for these guys was between 60 to 90%. So at this site, I think it was around 90% of all the animals that lived here died. We were super interested as scientists to understand how this loss of a keystone predator, the top predator in the ecosystem, which was like continental scale within a year or two of each other, affected the community. And I mean, it's pretty rare that as scientists, we get to watch how an ecosystem changes due to the loss of a top predator when we have a before picture, a during a picture, and an after picture. And one thing that we're really focused on is how ecosystems are resistant or resilient to that change. And so we're able to gain insights into how all ecosystems work. We have an ability to use the things that we find here and infer what's happening in a national park in the middle of America or a coral reef in Indonesia.
All right, welcome back, everybody. That was great. We're going to wrap up with the Bodega Marine Lab on the Bodega Marine Reserve. So the Bodega Marine Lab sits on the Bodega Marine Reserve, a 616-acre reserve located in West Sonoma County in Northern California. It's a stunning location surrounded by replicate rocky intertidal benches and with access to nearby kelp beds, urchin barrens, and the seagrass beds of Bodega Harbor. Incidentally, it's also where I did my PhD, so I might be a bit biased, but it really is a marvelous place. The Bodega Marine Reserve's mission is to contribute to the understanding and stewardship of the earth. Today, we're going to be hearing from Dr. Eric Sanford. Dr. Sanford is at the University of California, Davis, and has been faculty at Bodega for 15 years, where he studies range expansions and the impacts of changing oceans throughout California.
Hi, welcome to Bodega Marine Reserve in Bodega Bay, California. My name is Eric Sanford, and I'm a professor at the University of California, Davis. I'm a marine ecologist who's interested in studying rocky shore communities here on the California coast. One of the best parts about my job is that I get to spend a lot of time outdoors in really beautiful places like this one. We live in a time when marine ecosystems are changing rapidly. Human activities are increasing the concentration of carbon dioxide in the atmosphere, which is making our oceans both more acidic and warmer. For example, here in the Bodega Marine Reserve, recorded mean ocean temperatures have increased by about 1.4 degrees Celsius over the past 60 years. But the effects of these changes on marine life are still poorly understood. Therefore, a lot of my research is aimed at the question, how are warming oceans affecting marine ecosystems? One of the simplest predictions about climate change is that as the planet warms, species distributions will shift poleward to track these changes. Here in the Bodega Marine Reserve, we have in fact seen evidence that the abundance of southern warm water species is increasing. For example, we've documented an increase in the sunburst sea anemone, Anthopleurosola. This species was completely absent here in the late 1970s, but has increased in our region since the 1990s. In more recent years, its population has really taken off in the tide pools here at Bodega. How can we tell that a population is increasing? In this case, the methods are pretty straightforward. We conduct surveys over time by stretching out a transect tape and counting all of the sunburst anemones along the same 60-meter section of rocky shore. Over the last decade, we've watched the number of sunburst anemones on this section of shoreline grow from 25 individuals to over 500. So what does this new arrival of the sunburst anemone mean for other species here on the reserve? Well, the giant green anemone, Anthopleuris anthogrammica, is a more northern sea anemone that is common in tide pools on the reserve. One possibility is that these two sea anemone species might now compete for space. Although sea anemones might look a bit like passive flowers, they're actually animals that can be surprisingly aggressive. Beneath their tentacles, some sea anemones have white club-shaped structures called acaragi that are packed with stinging cells. Two undergraduate students here at the lab, Hao and Seth, conducted experiments where they put the two sea anemone species together in a laboratory tank. When the sunburst anemone is placed next to one of its northern counterparts, it inflates its acaragi and reaches over to repeatedly strike its opponent. Here you can see these interactions sped up in this time lapse. When they ran replicate trials like this in the lab, the sunburst anemone was always the winning aggressor over the northern species. Therefore, as these southern anemones increase in abundance here at Bodega, they might displace northern sea anemones from some tide pools through this aggressive competition. But the effects of climate change on marine ecosystems are not just limited to changes in average temperature over many decades. Instead, we're also interested in how extreme events like marine heat waves might be impacting our oceans. A marine heat wave is a period lasting from a week to more than 6 months where water temperatures are far warmer than normal. From 2014 to 2017, the West Coast experienced a series of marine heat waves associated with oceanographic events known as the warm water blob and the El Nino. These events led to extended periods when ocean temperatures were often 2 to 4 degrees Celsius warmer than normal. So what effect did these marine heat waves have on the distribution and abundance of marine animals along our coast? Well, our interest in this question began right here on Horseshoe Cove Beach. One morning in July of 2014, my collaborator Jackie Soanes was walking along this beach when she spotted a large, colorful jellyfish washed ashore. This turned out to be a purple-striped jellyfish, a southern species that is not normally found here in Northern California. Over the next few years, as the marine heat waves progressed, we documented many other unusual southern species arriving here at Bodega. To do this, we spent a lot of time in the field conducting surveys and looking for southern species that have not typically been found in our region. These warm water visitors spanned a whole diversity of different types of animals, including pelagic snails, nudibranchs, sea cucumbers, barnacles, and many other marine animals. We even found 19 pelagic red crabs, a species that is normally found swimming off the coast of central Baja California, Mexico. We kept these unusual crabs in seawater tanks here at the lab, and a few are still alive today. Many of these southern species disappeared from Northern California in 2017 when the marine heat waves ended and ocean temperatures returned to a more normal level. But some of these southern species can still be found here. A good example of a southern species that got a foothold here in Northern California during the marine heat waves is the owl limpet, Laudia gigantea. These large intertidal mollusks are herbivores. They crawl around at high tide, grazing diatoms off wave-splashed rock within permanent territories that they defend from other intruding limpets. Owl limpets are a southern species that was very rare here at Bodega before the marine heat waves began in 2014. But during these warm water events, we had lots of little owl limpets arrive from farther south. As part of their life cycle, owl limpets produce tiny larvae that swim along cilia. They look a bit like underwater helicopters. These larvae are dispersed by ocean currents and spend 5 or 6 days out at sea before they wash back to the rocky shore, where they attach and metamorphose into very small owl limpets. Every year we conduct annual surveys within the reserve where we count the number of limpets present and record their sizes to examine the survival and growth of owl limpets. In this way, we can determine whether owl limpet populations are increasing here through time. In addition to this ecological question, we are also trying to test what role evolutionary processes might play during geographic range expansions. In particular, we're interested in whether these new owl limpet populations here in the north might differ genetically from those farther south. It is possible that as owl limpet larvae were transported north, the larvae that survived this journey were only a specific subset of the genotypes that are found farther south. In other words, natural selection during dispersal might favor the survival of only those genotypes that are well suited to living in these northern waters. To test whether these evolutionary processes are occurring, we're collaborating with Dr. Rachel Bay here at UC Davis. Dr. Bay and her lab will be sequencing DNA from owl limpets to test whether these new northern populations have diverged genetically from southern populations. Sometimes people ask me, how did I first get interested in marine science? Well, my passion for marine life actually began by taking field trips during college courses. In fact, I even had the opportunity to take a field trip here at Bodega Marine Reserve over 30 years ago. Although it's not surprising that I've changed a lot over this period, what is surprising is just how rapidly our oceans have changed. We still have a lot of work to do to understand how warming ocean temperatures are affecting marine ecosystems, but our research so far indicates that southern species are increasing here in Northern California. In our future work, we're going to look at how ecological processes like changing competition and evolutionary processes like selection and adaptation might continue to influence how marine communities respond to climate change in the future.
Fantastic. Thank you, everybody, for those wonderful videos. I am going to invite the panelists now to turn on their screens and their audio. And we will get ready to start our panel discussion section. We already have some really great questions coming in. I encourage you to all keep putting those questions in the chat. Students, participants, anyone who's here, please keep those questions coming. Okay, our college classes have a time constraint of 50 minutes. So we're initially going to give priority to those questions. And after that, we'll answer questions from other attendees until the top of the hour. As a reminder, students, please do type the word student before your question and make sure your full name is visible. The panelists are going to try and keep their answers to a minute or less, even though they're very excited, so that we have a chance to get to some of these really great questions. Okay, to get us all started, I'd like to hear from all of the panelists briefly on the same question. Let's start with Jen Dijkstra, then Eric Sanford, and end with Silke Bachhuber. Remind us, what drew you to ocean climate change research and fieldwork in the first place?
Oh, for me, it was because I was doing my dissertation here in the Gulf of Maine, and along the way, as I was looking at all the communities, and I was actually in the years that I was doing my PhD, seeing real-time changes, and those are and then wondering why we were seeing real-time changes, or why I was seeing real-time changes, and then kind of correlating those changes with ocean warming. And so that's how I became really interested in it.
Yeah, great question. Thanks everyone for coming today. So I think I'm interested in fieldwork because I enjoy being outdoors and I'm fascinated by natural ecosystems. And I first got interested in climate change research when I was a grad student. And I got interested in studying how changing ocean temperatures were affecting the ochre sea star, which is the sea star that Sarah Graham featured in her video today. And like Jen, I think as my career has progressed, it's really difficult for those of us who study marine life not to take notice of climate change and how it's affecting these marine communities right before our eyes. And so that has led to a continued interest in understanding how changes in temperature and ocean chemistry and other aspects of the oceans are affecting marine ecosystems.
Yeah, I'll echo Eric's comment. I actually had the opportunity to participate in a research project as an undergraduate student looking at the effects of ocean acidification on larval abalone. And I had been super stoked about tide pooling and loved being out in the ocean, and was interested in marine science, but my interest in climate research was really piqued by that initial project and some of the work I was able to do as an undergraduate student after sort of getting started with that. And yeah, I'll echo Eric's comment that I think a lot of ocean research now and ecological research is climate research, and there's a huge amount of overlap in terms of what we're doing is all in a climate context.
And Silke, go ahead and take 15 seconds to introduce yourself.
Yeah, thanks, Kerry. Hi, everybody. I'm Silke. I'm a graduate student in the same lab as Sarah. And so we've worked pretty closely together on a number of different projects, including some of the ones that she described in her video. So she wasn't able to join us today, but I'm here as sort of her stand-in to answer any questions that you might have.
Thank you so much for being here. So let's get to some of these student questions in this 10 minutes that we have. Okay. I believe this question is for you, Silke. How are water chemistry changes that are occurring because of climate change affecting the predator-prey relationships of the sea stars and mussels? Have you noticed any changes in mussel shells getting thinner or thicker that would allow the sea stars to be more or less efficient predators?
Yeah, that's a great question. Thanks so much, Ben. And I'm going to answer briefly and then actually turn it over to Eric because this is also very much in his wheelhouse. But we aren't specifically seeing changes in the thickness of adult mussel shells right now in terms of the monitoring that we do. But there certainly are a number of different predicted impacts, both in terms of how ocean acidification might affect Pyzaster, as well as how it might affect mussels, a couple of which you mentioned, where they might become less abundant and have thinner shells. It's more energetically expensive for them to calcify and create new shell material as acidification increases. But there are also some really nuanced roles that things like local adaptation to variation in current pH conditions that we see along our coastline that can affect sort of some of those predicted responses. But yeah, Eric, I'll let you take that from there.
Oh, I think that's actually a great response. So there's a lot of interest in how changing ocean chemistry might impact predator-prey interactions. And Silke talked about sea stars and mussels. Another interaction that people have been interested in are predatory drilling snails and mussels, because these are snails that use their file-like tongue to drill a hole through the mussel shell. And if ocean acidification is causing mussel shells to get thinner, then those defenses are weaker and it might be easier for these predatory snails to drill through those shells. So it's a great question, and it's an area of active research. People are trying to figure out not just what are the direct effects of changing temperature and pH on organisms, but how are these changes also going to influence the way species interact with each other? So will things like mussels be more vulnerable to predators, for example? And yeah, it's still a lot of research going on that topic, so it's still kind of an open question.
Well, we have another question about predators, this one for Jen Dijkstra. What variables do you consider, such as predators, when monitoring every year?
So when we monitor, we actually, we collect photos of the seafloor, but we also collect underwater video footage of fishes, so that our fish community, as well as all of the larger invertebrates that I showed, lobsters and crabs, and sea stars. And those are I would say the vast majority of our predators.
And I guess other conditions that might disrupt your data each year that you come back. I think predators.
Sorry, I didn't quite catch
that. Oh,
that.
I'm sorry. One example was predators, but other variables that might get in the way of your monitoring efforts, I guess, is the question.
Oh, I see. If, for example, if the seas are really rough, then we're obviously going to postpone our monitoring. If visibility is pretty poor, so the Gulf of Maine doesn't have the greatest visibility, and so underwater visibility. And so if we have a large storm event or something like that, then we pretty much just hold off until the water clears.
Thanks. This question is for Eric Sanford. The owl limpet story is fascinating. Are they changing the face of the intertidal? And if so, how big is the impact?
Yeah, that's a really great question. So owl limpets are really important grazers on the rocks, so they feed on the algae. And one of the interesting things about owl limpets is that they are territorial, so they set up home on a little patch of rock and they spend their entire lives basically gardening and grazing the diatoms on that patch. And so, so far in Northern California, owl limpets have yet to reach a really high abundance. They're still pretty uncommon, but if their populations continue to increase, we would expect to see stronger impacts on rocky shores. So right now, owl limpets have a really major effect on rocky shores in Central and Southern California. And so if our numbers increase in Northern California, the prediction would be that, also those effects up here in Northern California will start to increase as they become more common. Excuse me.
Jen, another question for you. How are you using the stitched together photos of the seafloor in your research?
So I use these stitched together photos to create this larger photo mosaic of the seafloor. So essentially, you can think of it as a seascape, essentially. And so, and I guess, so using the seascape, we try to determine how invasive species are changing our landscapes or seascapes, and also how do our fish populations or other species utilize that seascape. So when you think about it as a forest, right, you can think that the forest is shrinking because our invasive species are a lot shorter than our kelp species. And because of that, it's changing our visual landscape. So you're going from a forested to a shrubland, and hopefully not a grassland, and then so when you think about how birds would then utilize that landscape, it's the same thing with fishes, how fishes are then visualizing that landscape to look for predators or to look for shelter.
That's a great analogy for someone like me who works in a forest. Thank you. All right, this next question is for Silka. If warming is affecting the upper limit of mussels and sea stars are hanging out in the water, what's the future for those sea stars even without the disease?
Yeah, that's an excellent question, and really kind of touches on a couple different areas of the complexities of ecological research, predator-prey relationships, and then gaining predictive insight out of that research when we think about it through a climate context. One of the things that we think about a lot in terms of climate affecting intertidal systems, especially rocky intertidal systems, is a concept known as a squeeze effect. And so if you have sea level rise that's potentially pushing up the lower limit of the mussel bed, as well as increased temperatures that are sort of pushing down the upper limit of the mussel bed. That's an example of a squeeze effect where that mussel bed is potentially getting thinner and thinner and thinner as temperatures increase, as sea level increases. We also see that Pythaster tends to forage kind of up until it's too hot for it to handle. And so as air temperatures increase, we might see decreased foraging behavior or extent of Pythaster. But the other kind of piece of that puzzle may be that as mussel beds tend to decrease or decline, we may see that Pythaster is able to change its behavior and potentially switch to other prey species as well. So Pythaster will sort of eat mussels as its preferred prey often until it kind of can't anymore, but then it may be able to eat some other stuff, or we may see a decline or in population or local extinction of Pythaster in some areas where their preferred prey, those mussel beds, are not able to persist. But there's sort of a lot of different things that can affect whether or not we continue to see this species in the fairly distant future as these changes occur.
Wow, everything's connected. So I am going to let the students go that need to go at 50 minutes past the hour, but I highly encourage you to stay because the next question is specifically interesting for students. But if you have to leave, thank you so much for attending today. The panel will stay on through the top of the hour, and we will be emailing a follow-up email with the questions, the answers to the questions we didn't get to. So you will get your questions answered. It looks like I'm frozen. I hope you can hear me. Okay, there it is. Okay, so the next question is, I'm actually going to have all of you answer it briefly. What advice would you have for a student who's looking into trying to get into the type of research that you do? And why don't we start with Eric Sanford?
Okay, great question. Well, there's always room for enthusiastic new scientists in this field. So that's an exciting question. I think taking relevant coursework is definitely an important first step. Building a solid foundation and understanding of biology and oceanography and marine sciences and all the relevant fields is really important. And then trying to get experience, if you're a college student, trying to get experience with other researchers. So if you have a professor who's teaching a class that you're interested in, you can talk to that professor about research that they might have going on in their lab. They may have grad students in their group or know people, and there may be an opportunity for you to get involved in helping with their research. And that's a really great way to expose yourself to marine science and gain experience and learn more.
Jen Dijkstra, do you have any advice for students?
I agree with everything Eric said. Yeah, I would absolutely recommend getting some experience, and as Eric mentioned, with the professor, with a grad student. So oftentimes professors may not have as much time as grad students to really delve into a particular topic. And grad students, they typically have a lot of, they typically need a lot of help. And so I would, yeah, Soko probably knows. And so, so I would actually recommend talking to the grad students and asking them whether they need help with anything, and being enthusiastic and getting to know not just your professors, but also the grad students, I think is a really good avenue.
Yeah, absolutely. Graduate students often need so much help. And I've been lucky enough to collaborate with a huge number of undergraduate researchers as I've worked on doing sample processing and some photo analysis and field work throughout the course of my research track for my PhD. I would tack on to emphasizing that the importance of getting involved in research as early as you can. And then also thinking about building those relationships with graduate students, lab members, kind of coming into it with a really curious and open mind, understanding that even if the work you're doing at the time doesn't necessarily mean that you're gonna be an intertidal scientist for your whole career, thinking about the main skills that you can gain from the work that you're doing and really using that to advance your interests and career as you move through your undergraduate career. I would also say, I would also recommend that you reach out to someone in your department, your academic advisor, someone in your college, as well as the researchers you might be interacting with, to see if you can apply for or have the opportunity to get funding to do a project. So we have a couple of undergraduate focused grant programs here at OSU. And I've actually had come up to 5 students that we've gotten funded to work together for an entire summer. And that was a really valuable research opportunity, an opportunity for them to gain experience, present at a conference, work closely with me on kind of parts of the larger project. And then that can also be a reduction in the barrier to access. If you're like me and worked through your undergrad, it's really challenging to have a volunteer position on top of that. So if there's funding available, definitely see if you can get involved with that as well.
Just to add a bit, I went to a big university as an undergrad and then a smaller university as a grad student, and it's a different world. So for students in maybe the bigger universities, you might need to be a little more persistent or get on a few listservs or go talk to people personally, but any research is, yeah, applicable across fields because the research experience itself, the field work itself is valuable. Great, thank you, everybody. Okay, we have so many questions. There's no way we can get to them all, but let's get to a couple here. Jen, your job sounds so cool. What challenges did you encounter along the way in school and after?
You know, I've been thinking about that question since it was posted. I've had a varied kind of career where in my undergrad, I studied terrestrial ecology, and I was on my way to becoming an ornithologist, bird person. And then, but I ended up kind of going to Germany and doing my master's in marine biology. And so I would say, and it took me a little while to kind of make that switch. So that was that to me was a bit of a challenge. I think too, the other challenge is just, quite frankly, just knowing what you want to do when you graduate. So I think for me, I have taken every opportunity that's been presented to me. I rarely ever say no to something unless I really don't have the time. And so I would say the challenge, I think my advice, and it's not really a challenge, but my advice would be to take those opportunities that are presented to you and just go with it.
Thank you for that. Okay, this is an interesting question because it links the research in Oregon and California. Since some of the Southern California species populations are moving up into Northern California, are the Northern California populations moving further north to Oregon? So I will let either Silke or Eric take that one.
I can start. So a lot of the northern species are ones that go from, say, Central California and currently go all the way up into Alaska. So I think a lot of northern species don't really have a place to go that's farther north because they're already in Oregon and Washington and have historically been in there. So I think what we would predict to see is that some of these northern species would start to become less abundant at the southern end of their range. Maybe in Central California, those populations would start to decrease. And there actually is some work on that. So there's a study that was done by Rafe Sagarin and some of his colleagues that resampled quadrats that had been sampled in the 1930s. So they were using basically the same sort of approach that Jen showed in her video, where researchers go out and sample everything that's present in a 1/4 meter squared quadrat. But in this case, the researcher from the 1930s had marked those points on the rock with brass bolts. And so the researchers were able to go back 60 years later and find the exact same quadrats that had been sampled in the 30s. And when they did that, they found that the southern species were increasing in abundance and the northern species had decreased in abundance over those 60 years. So that really highlights the importance of the sort of long-term monitoring that Jen and others are doing to sort of track how natural populations are changing. And yeah, the prediction would be that southern, more warm adapted species would increase in abundance and northern, more cool adapted species would decrease. And that does seem to be the general trend that researchers have seen so far.
And Silke, if you want to add like a 30-second extra.
Yeah, Eric really covered it. We're not seeing some of those sentinel species that he mentioned, like the Hopkins rose or Anthopleurostola necessarily yet, but we're definitely, our lab is involved in a number of long-term research projects where we are monitoring those shifts in abundance, not quite on the same time frame as Rafe's project was able to do, but we have some 20 to 30 year data sets looking at community structure like Sarah described that do allow us to understand how seasonal and decadal changes, as well as some of those shifts are potentially affecting community structure in the intertidal here in Oregon.
Okay, last question for all of you. Make it a quick one. What is one of the most challenging or most rewarding aspects of your job and research? Saw the eyebrow raise. Whoever wants to answer first.
Well, I would say one of the most rewarding for me is and exciting is being able to go out and collect data I really enjoy field work. And so I feel very fortunate to be able to do that on a fairly regular basis. And the most challenging, I think the opposite end of that spectrum would be, sitting at a computer and data crunching and analysis and writing papers. And that's also, a very important and good part of my job. But I would say that's probably one of the challenges.
I can go next. So I certainly agree with Jen that being able to do field work is one of the great joys of my job. And I'll just highlight one other, which is being able to work with students is really exciting, gratifying. So I'm fortunate to have a lot of really smart and enthusiastic students in my classes that are really passionate about marine life, and that really gives me great hope for the future to see this next generation of marine scientists coming along that have so much interest and concern and passion for our oceans.
Yeah, where I'm at in graduate school right now is I'm working on finishing up and writing my dissertation. I think that there's a duality to this sort of incredible frustration, but incredible stoke that comes out of taking the data that you have or the data that you're able to gather about these incredibly diverse, complex ecosystems that are changing all the time and have so many different things that affect sort of the community and the species that you see out on the rocks. And so every day I like sit down and battle to kind of wrap my head around the complexity of this incredible ecosystem that I study. And it can be so tear your hair out frustrating. But when you are in a situation where you make a graph and you look at it for an hour and then you're like, whoa, wait, hold on, that's insight. That's something really, really cool that nobody knew before that I now have documented and can share with members of the public and other people in my scientific community. That is both one of the most challenging components and one of the most rewarding components.
All at the same time.
Exactly. Yeah, it's a roller coaster.
It has been such a joy speaking with you three today. Thank you so much to our presenters and panelists, Silke Bachhuber, Jen Dijkstra, Sarah Graven, Eric Sanford. Thank you. We also want to thank the Organization of Biological Field Stations, the Center for Environmental Inquiry at Sonoma State University as well. I have links to those in the chat right now. Learn more about the series of virtual events at thevirtualfield.org and cei.sonoma.edu. And thank you also to the National Science Foundation for funding this project and all the wonderful people behind the scenes who made it possible. We will be emailing all of you in the next few days with follow-up answers to the questions we didn't address, a link to the recording, and a link to a very short survey, how you used the event, what you enjoyed, other event themes you might be interested in, suggestions. And this is to help us find funding and improve these for the future. And we're going to put this in the chat as well, this link. And thank you so much for taking the 2 minutes that it takes to fill that out. We hope to see you at a future Live from the Field event. We are currently planning the fall 2022 events. Dates and titles will be posted around July on the Virtual Field page and the Center for Environmental Inquiry page. These websites are also where you can find the recordings of all our past events. This is the 12th one now. Links to the websites for each of the field sites featured today and event resources. So everyone here took a lot of time to gather documents related to their work and papers you can read to get your classes excited about this before watching the recording. So please go ahead and take a look at those resources. Please also share this with your fellow instructors, students, friends, and colleagues. Thank you. Goodbye from the field.